Good day, everyone. My name is Joy Concepcion, and I will be your moderator today. On behalf of QPS's seminar series, I'd like to welcome you to ligand binding or mass spectrometry. That is the question. A few housekeeping comments before we begin. Everyone will be muted during this presentation. Please enter any questions under the control panel. If we cannot get to your question, we will respond to you directly after the seminar. This webinar will be available on demand to all registrants. We will send you an email once the recording is available. And now, I would like to introduce our presenters, Dr. Fabrizia Fussetti and Dr. Monique Putman. Fabrizia obtained her PhD in molecular biology and biotechnology at the University of Milan. She has over 50 peer-reviewed publications in the applied research of proteins. Fabrizia has been at QPS since 2014 and is currently the Scientific Director of Bioanalysis. Her main focus at QPS has been the development of mass spectrometry methods for bioanalysis of recombinant proteins, antibodies, ADCs, peptides, and oligonucleotides. Monique Putman obtained her PhD in microbiology at the University of Groningen. She has been at QPS since 2002 and is currently the Director of Translational Medicine at QPS Netherlands, where she leads PK and immunogenicity groups in the method development, validation, and support for discovery, non-clinical, and clinical studies of protein biotherapeutics. Please welcome Dr. Fussetti and Dr. Putman.
Well, thank you, Joy, for this kind introduction. I think, Monique, you might be on mute. Would you like to check?
Thank you. The problem is solved. Sorry.
Okay. So, go ahead.
So, good morning or good afternoon, everybody, and welcome to our fourth webinar of this year.
Well, hi, everyone. It is a pleasure for us to be here today for this open discussion and to share with you what we think are the key aspects when facing the challenges that are related to large molecular bioanalysis. All biotherapeutics, from monoclonal antibodies to peptides and oligonucleotides, have a very large degree of complexity and heterogeneity, and clearly also a large mass compared to small molecule. For many years, ligand binding assays have been the only option for large molecular bioanalysis. However, as mass spectrometry methods are more and more recognized as a valid and sometimes the only alternative, it is important to make some preliminary consideration of the scope of the project and the biology involved. I think selectivity and sensitivity requirements are often the most relevant aspects that drive the decision, but these are followed by other criteria, including timelines and regulatory aspects. The goal is at the end to obtain a method that is fit for the scope of the project and that provides a robust solution. They should also be compliant and time and cost effective at the same time. And well, you have equal access to both platforms. I think we should not be biased by the use of one or the other or prior know-how, but really let the molecular aspect and the pharmacology driving the thinking process. But also, beside these experts that are also related to the mechanism of action, the availability of reagents as well as the time that is needed to obtain those reagents during drug development can be relevant aspects to be considered. Monique is our Ligand binding expert, and here in Groningen, and we work together often to objectively define the optimal strategy that fits the project requirements at the start of the project, but also sometime during while a project is already ongoing.
So we start with a short introduction on ligand binding assays for those who are not very familiar with it. Ligand binding assays depend on a specific binding of antibodies to antigen or molecules to its target or receptor. And most assays depend on two binding steps as presented here. Samples are typically diluted and then immobilized using a capture reagent. And after a wash to remove all unbound reagents, a labeled detection reagent is added to the reaction mixture, and a complex can be formed. In a subsequent step, by choosing the suitable substrate, a signal can develop, and that can be either a colorimetric signal, a fluorescent signal, a luminescent signal, or even an improved sensitivity by applying a PCR step at the end of the reaction. Traditionally, ELISA makes use of a 69-well microplate reader, but over time additional platforms have been introduced, and nowadays several different options are available to perform the ligand binding assays. Most important for ligand binding assays are the capture and detection reagents. In some cases, these may be commercially available, but often these need to be generated specifically for the drug molecule. Monoclonal antibodies are mostly used, but FAB fragments or polyclonal antibodies, as already mentioned, target or receptor proteins may equally be suitable to set up a ligand binding assay. Based on the available reagents, different assay formats can be selected. And although most assays indeed use two type of reagents, it's also possible to develop a ligand binding assay based on one reagent, and that would typically be an inhibition assay. For ligand binding assays, samples are not extracted, but they're typically diluted to overcome matrix effect or interference by matrix compounds. And finally, the sensitivity can be affected by selection of the substrate and also the equipment that is used for the ligand binding assays. Typical sensitivities that are found are in the range of picogram per ml or low nanogram per ml.
Well, sensitivities will be often coming back in this presentation, as it is one of the major aspects that we consider during our thinking process. For mass spectrometry, differently than for ligand binding assays, high sensitivity can be achieved by carefully optimizing the sample preparation, which is a little bit more complicated than or laborious than a simple dilution for ligand binding assays. Essentially, the method starts with the extraction of the sample, followed by proteolysis for masses that are exceeding the limit of the instrument. After the proteolysis, the peptides are separated by chromatography. Sometimes they can be additionally separated by another chromatographic step. So you have like a two-dimensional chromatography. But eventually, in the mass spectrometer, there will be the peptides will be ionized and produce a measurable response. And it will be possible also to select specific peptides. The quantification will be in the form of area under the peak. So you will collect data from chromatograms. The addition of a representative internal standard at the known concentration in all quantitative mass spectrometry methods allows monitoring of the whole sample prep and often results in better linearity and accuracy of the assay. From all the generated peptides, one is chosen from each protein of interest, including the internal standard. And together with these other peptides can also be taken along for troubleshooting and can be very useful to derive additional information on stability or metabolism. There is essentially not a limit, or you can take many peptides in an assay. You can measure many peptides in an assay, which determines also the multiplexing ability of the mass spectrometry method. Steps can be omitted depending on the target molecule and scope, so it is not always necessary to go through the whole workflow. In a hybrid assay, the methods are essentially identical. It's just that the extraction step is achieved by using biochemical methods like affinity purification, most often on magnetic beads, or immunocapture. The reagents can be generic or specific. In general, the more specific, the higher the sensitivity of the assay. And the aim of the whole workflow is to increase recovery, reduce the complexity and the background to achieve maximum sensitivity and eliminate, therefore, all the possible interference from the background. The choice of the surrogate peptides will determine the selectivity and the ability to multiplexing, as I said before. And the assumption is that the concentration of the peptide, of the surrogate peptide, is stoichiometrically equivalent to the concentration of the target protein. So we do not measure directly the protein, but the peptide that is derived from the target analyte. From a mass spectrometry point of view, this is just a mass spectrometry method with a functional structural affinity purification. But from a ligand binding assay point of view, this could be considered an ELISA method with MS detection. Selectivity is often quoted as the main strength of mass spectrometry methods, and I have repeated already many times, I realize. And this certainly applies to hybrid methods. The first level of selectivity is defined by the affinity purification and the choice of the surrogate peptide. There is a second level of selectivity at the chromatographic level, and as this is able to distinguish between different analytes, but the maximum selectivity is probably achieved in the mass spectrometer that measures very specific masses and can define also, can discriminate also between very small differences and produces fragments that can be quantified. It's then able to identify very small differences like post-translational modification or metabolic change. The selectivity can be improved by using mass spectrometry with a high resolution. And well, as I said, the multiplexing capability is definitely an asset when using mass spectrometry. But this is probably not the only feature that distinguishes mass spectrometry from ligand binding assay. There are more, and Monique, I think I will guide you through this table.
Yes, as just discussed, selectivity is a very strong point of LCMS. And for ligand binding assay, it's the sensitivity that stands out. But of course, these are not the only characteristics that can be considered when selecting a suitable assay for your program. Ligand binding assays measure the entire molecule and is not limited by mass, whereas for LCMS, often a digestion is required and you follow a surrogate peptide instead of the entire molecule. ELISA assays typically have a limited dynamic range, whereas for LCMS, the dynamic range is much longer. However, by using other platforms for ligand binding assays, such as MSD, a longer dynamic range can also be established. For ligand binding assays, samples are not extracted, and you can suffer from interferences by matrix compounds, whether the workflow of LCMS is optimized to eliminate these matrix effects. Further, Ligand binding assays allow for higher throughput, and it's a very cost-effective analysis. So these things are really technique related, and in addition to these, you can also consider the requirements in your program. For example, is there a need to discriminate between free and bound drug, which may be easier to do with ligand binding assay. Further, ligand binding assays rely on the biological interaction between drug and the assay reagents, and can be set up such to provide information on the functionality of your molecule, whereas cyclic peptides that are measured with LCMS will not tell you whether the molecule is still functional. For ligand binding assays, you should bear in mind that anti-drug antibodies present in the sample may interfere with the PK assay, where for the extraction that is performed for LCMS will typically get rid of those potential interferences. And finally, you have to consider whether your program requires distinction between very similar molecules. For example, if there's a bond mutation or there are metabolites expected, these can be identified using LCMS, where most ligand binding assays will see them as a total sum of molecules. Overall, we see a lot of use of binding reagents, either for the ligand binding assay or for the sample preparation of the LCMS assay. And then says you can question whether these two are actually comparable. Importantly, when selecting an assay for your program, you need to consider what needs to be measured. So what is needed? What questions would you like to answer with the analysis that are going to be performed? And in addition, what can be measured? So are there any restrictions in your program or with the techniques that drive you into a certain direction? And this is essentially a decision tree. So we have pictures this here. There are questions that you can ask in which part of the program are you, preclinical or already clinical? What is the required sensitivity? Is there any cross activities expected? And these will guide you to a certain technique. And in all cases, for ligand binding assays, the main question is, are there any drug-specific reagents available to set up the assay?
Well, I will take over from here, going to different case studies. Well, probably the largest class of biotherapeutics nowadays is mainly constituted by antibody-based therapeutics. Here you can see the variety of IgG-based therapeutics that can be generated by building different specificity on a common scaffold. Well, it is a rather complex picture, and it shows how you can add different features to the conserved part of an antibody to generate chimer with multiple specificity, but also fragments that maintain the same specificity. Well, if you think about a hybrid LCMS method and the ability to multiplexing, but also the fact that you can easily use one generic reagent and then define the selectivity of your method afterwards, with the LCMS method, we can think of developing one single method for all different antibody-derived therapeutics, and especially if you consider IgG1 and IgG4 derived therapeutics. Indeed, all immunoglobulin are almost identical within one species, and sometimes that can be a clear advantage from a bioanalytical point of view. To reduce immunogenicity, humanized therapeutic antibodies are built by CDR grafting, which results in molecules that are practically identical to human IgGs, except for the antigen binding site on the variable chain, which will be developed to bind a specific receptor or ligand. Some regions, therefore, will display 100% sequence identity, and other regions around the antigen binding site will be completely or sometimes just slightly different. Depending on the need of designing a specific or generic assay, we will concentrate on either one or the other part of the protein. And this is possible with mass spectrometry, because after digestion of humanized antibody, there will be surrogate peptides that are common to a large family of IgG1 and IgG4 derived molecules. But at the same time, if you in the future or for a different purpose want to have a multiplexing assay that is able to quantify more antibody in one single method, it is possible to concentrate on peptides that are generated from the CDR CDR region, like I showed in this slide. In blue are the conserved peptides, and in orange and red coming from the CDR loops. When thinking about the bioanalytical strategy for humanized antibody, as a CRO, we often, if we are approached by a client for a project that is already in clinical development and the reagents are already available, then the ligand binding assay will be the most straightforward solution. But for preclinical analysis, our advice would be to use a generic hybrid method, as the sensitivity requirements are not very challenging at this point. And it will be possible to just develop one method for all IgG1 and IgG4 derived molecules, use a commercially available internal standard, and in this way, streamline the timelines that are often delayed by the need for specific reagents and for ligand binding assay. And also, so we already have thought about an assay that can easily be applied to all IgG1 and IgG4 monoclonal antibodies. And it is based on a magnetic beads purification with either protein A or L, or an anti-Fc antibody, depending on the type of molecule, if it is a fragment or if it is a full-length antibody, and then uses trypsin digestion, and then a selection of a surrogate peptide from the conserved region and the surrogate peptide from the commercially available internal standard. If we use a peptide from the conserved light or heavy chain, we will be able to use this method for many different many different antibody in different matrices. This method could even be applied for antibody drug conjugates, for instance, and to measure the amount of total antibody. In fact, ADCs deserve a special place as they demand on more complex assay setup for PK evaluation. And I think Monique is going to go more in detail into this from now on.
Okay. So antibody drug conjugates are therapeutic antibodies to which a cytotoxic drug or another effector molecule is attached. These are typically very potent molecules, and high sensitive assays are required. For in the preclinical area, both LCMS and ligand binding assays can be applied, but for clinical studies, typically the sensitivity that is required triggers a ligand binding assay. Of course, reagents need to be available, and in a program that we supported, we had the access to anti-idiotypic antibodies, and with these, a stepwise ELISA of ACLI assay was developed. In fact, for an ADC program, multiple assays are required to follow the PK profiling, so that includes total ADC, the conjugated form of the ADC, as well as the free payload. With the anti-idiotypic antibodies that we had available, very comparable assays were set up to measure total ADC and conjugated ADC. In fact, in both assays that we developed, the detection reagent was the same anti-idiotypic antibody, and the distinction between the two assays was the capture. For the total ADC, the capture was based on a capture reagent specific for the antibody part of the ADC, whereas for the conjugated ADC, the capture reagent was based on a reagent against the warhead part. In the end, these follow a sandwich, and an ECL signal is generated once a voltage is applied to the plate. Both assays had been thoroughly developed and validated, and they really work nicely. As you can see here from the validation data, both assays had a long dynamic range, and the sensitivity for the conjugated assay was somewhat better than for the total assay, but both were very suitable to support the program. And the precision and the accuracy of these assays were actually very nice, and that is largely depending on the reagent's quality. And the numbers that we found are very comparable to precision and accuracy that you actually also see for LCMS assays.
Well, I will continue from now with another case study where the bioanalytical strategy was practically already defined at the beginning of the project, as it was the only possible solution. This happens often with therapeutics for replacement therapy and sometimes also for gene therapy projects. The scope of the project was to develop a selective and multiplexing PK assay for two proteins that were practically identical and should be simultaneously quantified in plasma and in preclinical species. One was a recombinant protein and the other one was an endogenous protein. Commercially available antibodies and ELISA kits were not able to distinguish between the two different proteins, and not even a functional assay could be applied. So even if the sensitivity requirements were rather challenging, the mass spectrometry, also based also because of the multiplexing ability, but mainly because of the lack of specific reagents, was the most suitable technique for this project. We were very lucky that these two mutations on the protein were located such that upon digestion, the proteolytic peptide generated would be different between the endogenous protein and the recombinant protein. And then by focusing on these two regions, we selected the two peptides that gave the highest sensitivity, and develop an assay based on using the commercially available reagents, but using then the selectivity power of the mass spectrometry afterwards. In the next slides, there are the validation parameters from which you can see that the assay, the assay is yet to be validated. It's now in the process of being validated, but these are data from the qualification, and you can already see that the quality of the assay is very high, but then we were even able to not only to meet the requirements of sensitivity that were around 150 picomolar, but even improve that by twofold. So we have here a sensitivity of 5 nanogram per milliliter, which for this protein corresponds to about 70 picomolar. The chromatography was the step in which we could also clearly separate the peptides from the endogenous protein and from the recombinant protein, as there was a clear retention time gap. But because of the mass difference, we would have been able to have independent quantification of these peptides, even if the separation wouldn't have been so good, probably in theory. And I think this data, maybe it's interesting to point out that this data was generated without the use of an internal standard, which it does not really affect the linearity of the assay. It is already very good, but we will make an effort to use a peptide, a label peptide, to be able to monitor and even improve the performance of this assay. And I think the next project is focusing on recombinant peptide hormone, where yeah, the discussion at the beginning of the project was rather complex, and I think Monique can continue with this.
Yeah. So it's indeed, and the last example concerns a recombinant peptide hormone. When this project was presented to us, there were a few things to factor in during the selection of the assay to be used. Antibodies against both the N and the C terminal of the molecule were available at the time, and a preliminary assay had been set up, but would require additional development. And the assay would be required for several species. The target sensitivity was 10 picogram per ml, and also it was questioned whether we could measure the entire molecule or also different metabolites or degradation products. So when we evaluated which assay was best suitable for this project in a preclinical space, we could not really decide straight ahead. So we decided instead to do a short feasibility in which both techniques were tested in parallel. So a week was spent both on liquid binding assay as well as LCMS. And for the ELISA, and a relative straightforward sandwich was set up with an MRD of 10-fold. And it was estimated at that time that the sensitivity could reach approximately 30 picogram per ml. From the preliminary LCMS experiments in which a protein precipitation was applied, it was expected that the limit of quantitation would be approximately 200 picogram per ml. And also the experiment showed quite some carryover, which indicated that method development would take quite some time. So based on these initial findings, it was decided to move ahead with the development of the ELISA. And already during the feasibility, we noticed that the reagents that were available did not have an optimal affinity. And in order to have maximum sensitivity of the assay, we decided to set up an homogeneous sandwich assay in which all the reagents are added together with the sample at the same time in a non-binding plate, allowing the complex to form and then transfer it to a binding plate, after which only one washing step is required before the addition of the substrate. And this assay worked well and was validated, but unfortunately during the validation it was found that at the lowest level the precision and accuracy was not adequate, and LLOQ had to be raised to 227 picogram per ml. For further studies, it was decided to optimize the assay, and the assay was changed to an MSD platform with a light signal rather than a colorimetric signal. And that was successfully validated for rat plasma samples with a limit of quantitation of 44 picogram per ml. And subsequently, the assay has also been used for monkey, rabbit, and mouse, and all work similarly. But by the time we wanted to use the assay for human, we unexpectedly saw a very high background. So you can see here that at lower concentrations in the curve, you see an assay signal of about 10,000 relative light units, and you lose most of the calibration curve because of that. Many experiments were performed to see what happened, and it was found that the capture and the detection antibodies could be linked by the presence of human IgG, which was very unexpected finding because it was nothing like the drug itself. And we did several tests to see if we can overcome this interference by either blocking the human IgG or extracting it from the sample, but we could not get rid of the background in sufficient way. And at that time, it was realized that we still had the possibility to use LC-MS instead. So as we had already gained some experience with using the available antibodies, it was decided to make a hybrid method in which an affinity purification was performed using the available antibodies that were also used for the ligand binding assay. And then the LC-MS detection. For this particular project, it was not needed to perform further sample treatment or digestion, and the sample could be directly measured. And the assay, in the meantime, has been validated successfully with a limit of quantitation of 50 picogram per ml and very good precision and accuracy. And this is an example of a project for which during the program, the transition of methods is required because of unexpected findings that occur. So, in conclusion, the starting point is what needs to be measured in your program and which questions need to be answered. So the project goal and the main constraints of the assay, these guide you to some kind of decision tree to the best assay for your purpose. As shown, the analytical strategy can change during development based on things that are encountered during the program, or you can also expect, for example, from the switch from preclinical to clinical, that the sensitivity needs to be improved, and that may trigger an analytical strategy change. And further, it is there are other things to consider, such as the time that is required to obtain reagents that are needed for the assays, make a realistic assessment, and also check what kind of regulatory requirements would be best for your particular project.
Yeah, I think as a last remark on this conclusion, I would like to mention that yeah, as more and more techniques are becoming available, because it is possible to analyze an even larger variety of molecules with optimal data. And there is not a perfect solution that fits all these challenges. Therefore, we think it's really important to focus on what are the relevant aspects and the molecular aspects of the target molecule, and then look at the mechanism of action, keep the biology in mind. We do not have a specific preference for one or the other technique, so we are not really biased towards using ligand binding assay or mass spectrometry, but we really listen to the requirements of the assay. We count on a multidisciplinary team, and that is all in one place. So this expertise is concentrated in one place, and we have a team of scientists that do not only have biochemistry or cell biology background, but also have affinity with methods like mass spectrometry. And finally, and I would say the last but not least, it is really very important to keep open communication between the drug development team and the bioanalytical teams within pharmaceutical companies, but especially when these two teams are located in at the developer side and at the CRO side, as this can be really determining the success of a bioanalytical program. And with this, I think, Monique, if you do not need to add anything to this, do I have a last remark? I would like to thank everybody for listening and for watching today. Thank you for your attention.
Thank you, Fabrizia and Monique. And now we're going to take some questions. The first question we will take is the use of an internal standard always needed in LC-MS methods?
Well, I think I can take this question. It is convenient to have an internal standard, and it will increase the linearity of the assay, especially if validated when we need to validate an assay. However, as I showed in the example of this recombinant protein, it is not always necessary to have an internal standard, as sometimes if the recovery is linear and there is no need, and the reagents are always behaving well, there is sometimes no need to include an internal standard. However, if an assay has to be robust for a long period of time, I would advise to use that, but that can be done in a second step during, in the, let's say, developing GLP methods.
Okay. And which platform would you use for candidate selection in non-clinical and early tox studies?
Monique, shall I take this?
Yeah, yeah. I would advise actually to use LC-MS as assay for that. So maybe you can give a little bit more detail.
Well, if Monique says that we can use a LC-MS assay, then I think, well, I agree with her, especially because in that respect, you can certainly use the ability to multiplexing if you want to monitor more candidates in one round. But you can also develop one assay that is specific for a class of molecules. And we have seen that, for instance, probably the ultimate class of molecule are monoclonal antibody. And in that case, it is possible to use a generic assay that is already developed and sometimes already validated to monitor all different antibodies with different specificity if they all have, if they're all humanized antibody, for instance. So then, yeah, I would say certainly LC-MS as the most time-effective solution, and also probably cheaper. We'll give you
I think also a point here is that during candidate selection, always or often not all the reagents that you would require for a ligand binding assay are available. So it will also save some time if you can start with the LC-MS analysis and not wait until reagents are available.
Right. Which platform would you use if you had to monitor drug concentration as well as receptor concentration? And can those be quantified in the same assay?
I think I can start with a small answer. I think both platforms allow for multiplexing. Not a standard ELISA, but there are other platforms that allow you to. But there's quite some method development to perform for a ligand binding assay to ensure that there's not a lot of interference mutually. So if you would like to do a multiplex analysis, LC-MS would probably be easier to set up.
Yeah, also again, because you can monitor more analytes at the same time. And if the dynamic range and the assay range for these two molecules is comparable, you can even think of doing analyzing them in the exact same assay.
Okay. And how can you distinguish free from total biological drugs using ligand binding assays?
So if you, for example, measure a molecule that binds to a particular target or receptor, then often certain binding places are occupied by that binding. So if you choose your reagents such that you have reagents that can detect the free form only, for example, by if there's a competition for that binding place with the receptor, then you will only measure the available and the available drug. Whereas if your reagents are directed against a part of the molecule that is not blocked by the functional binding to either a receptor or a target, then you can measure the total amount. So by the selection of the setup of your assay, you can select what you would like to measure. In addition, you can apply some acid dissociation, for example, to make sure that you measure the total amount and not only the free amount.
Right, so that it's not underestimated?
It can be underestimated if you are not paying attention to what is actually measured.
Gotcha. What is the percentage use of hybrid methods in full GLP compliance context? So, for example, preclinical GLP tox studies, considering that there's no guidelines exists for acceptance criteria.
Yeah, well, the discussion on the acceptance criteria is really ongoing since hybrid mass spectrometry have made their appearance in the biology in the large molecule space. But we see that most of the time you can validate an assay with using the criteria for chromatographic methods, especially if your internal standard is really representative. And if the assay is carefully optimized, and if you can also use automated steps to reduce the manual error, you can definitely come down to lower than 10% differences for precision and accuracy and relative error determination. I think in percentage, I don't know. I cannot now give you a number, but we see that it is more and more used, especially because of the fastest timeline. And I think if you can understand also the meaning of the criteria for the acceptance criteria for your molecule, it is also easier to justify the use of maybe higher criteria, more similar to larger molecule, but more similar to the ones that are used for large molecule bioanalysis, because they strongly depend on the type of reagent. At the end, from that point on, you will have a normal mass spectrometry method. The variability will be in the affinity purification. So if we consider that equivalent to a ligand binding assay, I think it's justifiable to use largest criteria, which doesn't mean that you are, like I've heard once, somebody saying, doesn't mean that you are pipetting upside down because you have larger criteria. We will still be looking at the performance of the assay and care for precision and accuracy and quality of the data.
Right, understood. You showed an assay setup for ADCs. Do you have any strategies to handle DAR sensitivities?
Yeah, for the DAR species, typically I would expect an LCMS assay to be applied.
Yeah, you need to use intact. Today we mainly focus on bottom-up approaches, which are now used more routinely in compliant regulated bioanalysis. For data determination and quantification, you would need to move to high-resolution mass spectrometry and look at the quantification of the intact molecules. Those methods are available, and I don't think you can have a validated, you can rely on those methods for a validated method, but they can certainly be used as qualitative assays for determining to quantify the different species, and they are certainly very informative for stability studies and yeah, to understand the dynamic and the pharmacokinetics of ADCs and safety assessment. I don't know if this answered the question. Can you repeat the question once more?
Excuse me?
Can you repeat the question once more?
What's the strategy for handling DARs since we've worked on ADCs before?
Yeah. So then high resolution mass spectrometry.
Okay.
With
can you see immunogenicity effects on PK if the data is determined by LC-MS?
Well, if you do not use a high width, if you do not use an affinity purification, that has to do with the functionality of the method. That you, for instance, you can use a receptor for purifying an antibody. Then you will probably see also an immunogenicity effect. If you use a straightforward LC-MS method in which you do not use a functional assay to purify the protein, then after digestion, you will identify the total protein, and that will be, and immunogenicity will probably not have any impact on the amount of protein present in the assay. So it all depends on how you design the sample preparation. But I don't know, Monique is an expert of immunogenicity assessment, so maybe she can add something to this.
No, I think I agree with your answer, because if there's any antibodies present and bind to your molecule which obscure the binding places that you use for the affinity purification, you will certainly have an effect.
Okay. And your case studies mostly refer to PK assays. What about biomarkers?
For biomarkers, you can essentially make the same type of decision trees and ask the question, what do you need to measure? Because whether biological is a drug or a biomarker, yeah, you can apply the same type of techniques. So you can have the same constraints and the same type of decisions to decide on the assay platform. Of course, there are many commercially available ligand binding assays for biomarkers that you can save time by using those if suitable for your program. But there's no need to exclude LC-MS for biomarker analysis.
Yeah, I think every time we consider that option, or let's say most of the time we consider that option, the sensitivity was probably the only limit to the technique. And if the sensitivity of the assay and is sufficient to determine the baseline, then I think you can use the two methods in equivalent way.
Okay. Well, that is all the questions that we have for today. I want to thank you both for your presentation, And I want to thank everyone for their time and attention. And this webinar, as well as others, will be available at the QPS website with the address listed on the slide there. And this webinar should be available usually by the end of the week or early next week. So again, we thank you for your time and attention, and we wish you all good health and a happy holiday. Thank you very much.
Thank you very much. Bye-bye.
Thank you. Bye.
Bye.